Welcome to the webinar series hosted by Anabios. My name is Richard Kondo, and I am Vice President of Business Development at Anabios. Thank you for joining us today on today's exciting talk by Dr. Ted Price on the mechanisms of neuropathic pain in the continuing series of presentations hosted by Anabios on the research developments in the area of pain. As the audience is well aware, Dr. Price and his group at UT Dallas has been elucidating the molecular profiles and associated mechanisms underlying peripheral sensory nerve function and of central mechanisms in the spinal cord. His group has developed novel approaches to uncover these profiles, and he will be presenting on new data that his group has gathered. Next slide, please. First, I'd want to thank Gary Watkins, Director of Marketing at Anabios, for putting together this webinar series. As a quick intro to those who have not joined us in the past, Anabios is a unique contract research organization and biotech company based in San Diego. We recover human organs from ethically consented donors and use the tissue or cells to conduct physiological assays in which we can test preclinical compounds for drug discovery projects. We have essentially redefined first in human. And working with Anabios is as close as you can get to a clinical trial without actually doing a clinical trial. For those developing therapeutics for pain, Anabios has been evaluating client pain therapeutics for over 15 years. Dorsal ganglia as well as trigeminal ganglia are recovered from postmortem donors with both normal and pain or neuropathy pathology. Anabios provides services for the entire drug development pipeline, from target identification validation through the provision of tissue samples for transcriptomics or proteomic work, or validation of sensory neuron modulation by tool compounds, to lead identification through supporting medicinal chemistry or evaluation of gene therapy constructs to lead optimization and candidate selection. Now, finally, a few technical points regarding the webinar. First, there are handout PDFs regarding Anabios pain-related products and services that can be downloaded from the handout section in the webinar control panel on your lower right. Regarding questions, we would like to hold them until the end of the webinar. To submit them, please use the chat box in the webinar control panel. You can type in the questions at any point, and I will read the questions at the end of the presentation. So without further ado, it's my great pleasure to introduce today's speaker. To many in this audience, today's speaker needs no introduction, as his lab has been revealing the secrets of sensory function in human for many years. Nevertheless, it's a delight to introduce Dr. Theodore Price. Ted Price is the Ashbel Smith Professor of Neuroscience at University of Texas at Dallas. He received his bachelor's degree in neuroscience from UT Dallas and his PhD in pharmacology from the University of Texas Health Sciences. He did his postdoctoral work at McGill University with Fernando Severo. His lab at UT Dallas investigates the molecular and cellular mechanisms underlying pain plasticity with a focus on how chronic pain develops and persists. Their goal is to discover novel therapeutic targets and develop non-opioid treatments that can prevent or reverse chronic pain conditions. Please take it away, Ted.
Thanks, Richard. You can hear me fine, right?
Yes, you are.
Okay, great. Excellent. Thanks everybody for being here. Richard, thank you for inviting me, and thanks to Anabios for the platform. What I'm going to do today is tell you about our work working with human DRG and spinal cord, and how we are using this really amazing resource to better understand mechanisms of chronic pain, in particular neuropathic pain. So our work has started with Anabios many years ago. I'm still extremely grateful to Anabios for the work that we did on our initial transcriptomic work on human DRGs. Over the years, we've developed a really amazing collaboration with the Southwest Transplant Alliance here in Dallas. And this slide shows what we do with them. So we do recoveries here at the Southwest Transplant Alliance Legacy Center, where they have their own operating rooms. And we routinely recover DRGs, which you can see here in this specimen that was recovered from an organ donor, and the spinal cord. And we do these recoveries usually about 2 to 3 times a week, and the tissue is recovered from the organ donors with a postmortem interval of between 2 and 4 hours, but usually closer to 2 hours. And we recover all kinds of tissues from these donors. We recover thoracic and lumbar, dorsal root ganglion, on occasion sacral DRGs. We recover the spinal cord, vagus nerves. We've done nodose ganglion recoveries. We're doing work on spinal meninges, peripheral nerves, and also in collaboration with Saad Yusuf's lab here at UTD, we've also been doing work on the sympathetic ganglia. And because we can recover these tissues with such short postmortem intervals, we've also been able to develop in vitro assays where we can record or do calcium imaging or signaling assays on dorsal root ganglion neurons. So we're routinely doing this kind of work in our laboratory, which again is very similar to kind of work that Anabios also does at their site. So I want to start out by thanking the organ donors and their families for the incredible gift that they give to us to allow us to use the tissues from their loved one for this research. And I also want to point out the really wonderful people that we work with at the Southwest Transplant Alliance. Aaron Vines is the research coordinator that kind of makes everything possible. And then Anna Cervantes, Peter Funk, Jeff Funk, and Peter Horton, Jeff Funk, and Tariq Khan are the surgeons that we work with on a regular basis to recover these tissues. So I'm going to talk about a mix of published and unpublished work today, but I want to start with a amazing project that I have been referring to as the Big DRG paper, which I hope will be an exciting preview for something that will be coming out here pretty soon. So this effort that led to this paper has been part of the Precision Human Pain Network. This is a NIH-funded group of centers through the HEAL initiative. And there are 4 centers. There's us at UT Dallas, which also includes Pat Doherty's lab at MD Anderson, and Michele Curatolo and Christoph Hofstetter at University of Washington in Seattle. There's another group led by Rob Giroux at WashU, a group at Harvard led by Will Renthal, and a group at University of Pennsylvania led by Wenchen Liu. And collectively, we have put essentially all of the work that we've been doing over several years now on dorsal root ganglia recovered from organ donors, fast autopsies, and relatively rare surgeries into a huge atlas of the human DRG, which I'm excited to say should be available as a preprint in about a week. That will depend on how fast bioRxiv can screen this, and all the data sets are set to be released by the end of this week, at least from our side. So what is in this paper and what have we found? So first of all, we have been doing across all the different centers DRG recoveries from a variety of different sources, and we now have, in putting this paper together, have massively exceeded the number of human DRG neurons that have been sequenced previously. And this also includes non-neuronal cells, but for neurons, you can see like we really have pushed the field forward really dramatically. The data set that we're about to release is from a total of 126 donors coming from across all of these sites, with a grand total of more than 50,000 neurons and about 500,000 non-neuronal cells. And importantly, and unlike any previously published atlases, this includes DRGs from all levels. So we have a representation of the lumbar DRG, the thoracic DRG, and the cervical DRGs. The one that is still underrepresented is the sacral DRGs. This is mostly because these DRGs are embedded in the pelvis, and it's very challenging to recover them from organ donors. We have a good even distribution of male and female samples. And we have samples from all different times of life from humans, starting with pediatric organ donor cases, which we started doing here in Dallas several years ago, all the way up to samples from individuals in their 80s or 90s. So we have a really expansive data set here. And just focusing on the neurons for the first part of the talk, we have now identified what we think are the definitive subsets of neurons in the human DRG. And we've identified now 22 transcriptomically distinct neuronal subtypes. And for each one of these subtypes, we have identified gene markers that can allow you to detect these subtypes using RNA-scope or spatial transcriptomic experiments. Sometimes there's a single gene marker, sometimes it's a combo of markers. You can see that we've also tried to define them as A fibers or C fibers. Some of the A fibers are likely nociceptive in nature. Some are likely not, for instance, proprioceptors and A-beta low threshold mechanoreceptors. So hopefully when people get to see this entire paper, when it comes out very soon, it'll be easy to understand what these are. And while I'm not going to go into it in the slides, we also have been able to equate these neuronal types to their pairs in the mouse. And the markers for these neurons hold up pretty well from mouse to human. And then we've used data sets produced by labs like David Ginty's lab to try to better understand the potential function of these neurons. So a lot of the details about that will be in the preprint when it's released. So as part of this, one of the main things that our group has really been interested in is understanding what are the transcriptome of each one of these individual subtypes of neurons, what are the G protein coupled receptors that are expressed, the ion channels, etc. And there is a bit of divergence from mouse. So I think, it's really important to understand the expression in these different human subtypes. And this data set really goes far beyond anything that has been published before. So we're really excited for the pain community, as well as just the sensory neuroscience community, to be able to use these data sets. And we can look, for instance, and see if there's differences in the representation of these data sets across different levels of the cervical, thoracic, and lumbar DRGs in humans. And indeed, there are some differences in the distribution of these subtypes across these different levels. For instance, there's a particular subtype of proprioceptor that is more heavily represented in the cervical DRG than in the thoracic and lumbar DRGs. And we think that this probably relates to muscle spindle innervation in the muscles of the neck, which we know has a different innervation pattern and a different number of muscle spindles in those muscles than are found in other areas of the body. So it's been really interesting to see how this has all kind of come together to fit with what we know about human sensory physiology. We also see some sex differences in populations of representation of populations of neurons, although these are relatively small, but you can see there are some significant differences here. And there's also some differences in representation of these populations between younger donors and older donors. And I can't tell you that we understand exactly what this means now, but I think it's interesting and gives us some clues to what happens to the dorsal root ganglion as we age. And of course, we know that as people get older, incidence of chronic pain increases. So there's probably some clues here into why that might be happening, in addition to what we already know about what happens to our lumbar discs and our joints as we age. that kind of covers the basic state in the human DRG. And again, I didn't go into a lot of detail on that because the preprint is about to come out, and I'm excited for people to be able to read that in a lot of detail. What I wanted to dive into in more detail from our lab's specific work today is how we're trying to understand what's happening in the human DRG in disease states. So here I'm going to talk about two projects. I'm going to first talk about our work on diabetic neuropathic pain, and then I'm going to talk about how we're trying to develop humanized models for target discovery in arthritis and low back pain. And there I'll focus mainly on low back pain. But first, the diabetic neuropathy story. So this is work that's been led by Stephanie and Aish in my lab. And we had a paper that was pretty recently published in Nature Communications that I'm going to go into a little bit of detail on in the next couple slides. So when we started doing our work on human dorsal root ganglion, and soon after we received our funding through the Precision Pain Network from NIH, we needed to develop very rigorous methods to do quality control on all of the human DRG samples that we obtain. So Stephanie has been leading this effort for the entirety of this project, and one of the key steps in her tissue processing is to simply cut sections and do H&E staining. So here you can see an example of this H&E staining, and all of these samples are very high quality samples, and essentially we've worked out methods for freezing tissues in the operating room to make sure that we have optimal preservation of the tissue for downstream analysis. So what Stephanie noticed when she was doing this is not all of these DRG samples looked the same. She was noting that in some DRGs, like this one here, where you can see it very clearly, if you look at the bulb of the DRG, you can see that there's regularly spaced large neurons, and I say large because all human DRG neurons are pretty large. Obviously, they're on a distribution where the C fibers are smaller diameter and the A fibers are larger diameters, but you can easily see how these neurons are usually spaced out within the bulb. You can see the same thing over here on this one. But as you move across the screen here to these ones on the right, where you would expect to see neurons there, you now see these accumulation of nuclei. And again, most of the time what you see is the nuclei are kind of spread out, and there's a ring of nuclei that go around all of the neurons, and those are the satellite glial cells. But here, there's a much different looking pattern, and on this one on the far right, it's like really extreme. You can see lots of these things. So Stephanie started noticing this, and being the very careful scientist that she is, she went about quantifying them and also looked in the literature and found what they might be. So turns out that they are Naget nodules, which I will tell you more about in the next slide. These were originally discovered by Jean Naget back in the 1920s, and they're named for him. Remarkably, what she found is that in most cases, DRGs where we see a large number of Naget nodules, they're coming from individuals with diabetes, and also likely people who have diabetic neuropathy and pain, because they have a medical history report of diabetic neuropathy, and they have a medication history that includes analgesics like gabapentin. So that's what's shown here in this graph on the left. In the center graph, we show how commonly these are occurring. And you can see in DRGs without diabetes, and we actually have a much larger sample size than what's shown here now. We don't see very many of them. We do see some of them, and they seem to increase with age and independently of disease. But in these individuals with diabetes and peripheral neuropathy, we really see a large number of them, and it can be as many as 30 to 40% of the neurons. And this is pretty important because these Naget nodules form when the sensory neuron degenerates, and then Jean Naget's original idea was that satellite glial cells proliferate and then kind of take over the space where that sensory neuron seemed to be. So this is actually an indication of degeneration of neurons within these DRGs. So the next question is, does this only happen in diabetes, or does it happen in other forms of peripheral neuropathy, or does it even happen in people just that have chronic pain from things like arthritis? So while the sample sizes here are small, the pattern is pretty clear. We see Naget nodules in individuals who have what we think is a history of neuropathic pain. So for instance, we have a couple of examples where individuals had painful alcohol neuropathy, idiopathic neuropathy, fibromyalgia, but were being treated with gabapentin. So, maybe it was misdiagnosed idiopathic neuropathy. But then if you look in individuals with arthritis, we don't really see Naget nodules in the DRG. So it really suggests that these Naget nodules are associated with neuropathic pain. So Naget nodules are not just a degenerating neuron and a proliferation of satellite glial cells into the area where the neuron degenerated. They also include this amazing sprouting from nearby neurons that kind of invades and forms these small neuroma-like structures within the DRG. So Naget first described this in experiments that he was doing in rabbits, where he was doing a experimental removal of the leg and cutting the nerve, and seeing this degeneration that happened within the associated DRGs after the amputation. So what Stephanie wanted to do is to look to see if we saw these similar structures in human DRG. And of course, back then, Naget didn't have the kind of imaging tools that we have now, so we could apply confocal microscopy to this. And what we see is just an amazing sprouting of neurons into these Naget nodules. And one of the things that we wanted to try to understand in this project is, are these coming from neighboring sensory neurons? Are they sprouting sympathetic fibers? What is the nature of these sprouting neurons? And at this point, we're pretty convinced that they come from nearby, probably C nociceptors. We see a large amount of peripherin in these nerve fibers. We also see really intense NaV1.7 staining. And of course, everybody knows NaV1.7 is associated with pain in humans from the genetics. And we also see NaV, although it's not shown on the slide, we also see NaV1.8 staining within these. And it's very reminiscent of what has been described by Steve Waxman and Suleiman Dibhaj and others, looking at neuromas at the site of an amputation, for instance, in humans, except remember, in this case, these are very, very small, and there could be hundreds, if not even thousands of them within the DRGs of these individuals with neuropathic pain, mostly diabetic neuropathic pain. So Stephanie also could apply tracing methods to look and see where these were coming from. And you can see an example of such an experiment here. So there's a nearby peripherin positive neuron here, and then there's a Naget nodule, which is shown here. And you can see that we can trace most of these axons back to this peripherin positive neuron here. Sometimes there are these axon blebs, and then there's another group of axons that comes out. So you can see here that these this kind of nest of axons over here forms a bleb, and then there's another nest of axons that comes off of them. So they're really amazing entanglements of axons here. And we think that it's at least possible that these could be sites of ectopic activity within the DRG, much like neuromas are known to be sites of ectopic activity that cause neuropathic pain in humans. So another very interesting observation from Stephanie's work is that these neurons take on a kind of morphology that kind of looks like what happens when you culture an adult dorsal root ganglion neurons from humans or from mice or from rats for that matter. So everybody knows that sensory neurons, DRG neurons, are pseudo-unipolar. But you can see that this neuron here does not appear to be pseudo-unipolar anymore. It seems that it's become multipolar with multiple axon branches coming off. So we've seen many examples of this where these neurons seem to really fundamentally change their overall morphology in this pathological state. So we don't yet know what the physiological consequences of them taking on this multipolar-like morphology is, but that's a key thing that we want to try to understand in the future. So there's much more detail about all of that in the paper that we published this last year. But one of the key questions that arose from it was, what is the sensory neuron population that is dying in individuals with diabetic neuropathy? So ICE wanted to try to tackle that question and has now done single-nucleus RNA sequencing from a fairly large number of control samples and also diabetic peripheral neuropathy samples. And what she's found when she has analyzed all this data, which is all together here on this graph on the right, is that there are specific populations of neurons that are represented in this UMAP only by non-DPN samples and not by DPN samples. So it doesn't appear to be the case that the neurons are just randomly dying. In fact, it seems to be certain populations of neurons within the DRG that are highly susceptible to cell death. So what are these populations? Well, it turns out that they seem to be A fibers, and one of them is a particular type of A fiber low threshold mechanoreceptor that we've been pretty interested in because it expresses the T-type calcium channel, CAV 3.2. So we've been working on CAV 3.2 expression in human DRG with Emmanuel Bornay's lab, and we published a paper in Brain on this also earlier this year. So what we see in the diabetic neuropathy samples is that this population seems to be falling out. There's also a major loss of CAV 3.2 gene expression. And we think this is pretty interesting for a couple of different reasons. So one is that CAV 3.2 inhibitors went into clinical trials for diabetic neuropathic pain a couple of years ago and didn't show efficacy. And based on the data that we have here, we wouldn't expect them to have efficacy because late in disease, there is very little CAV 3.2 expression. In fact, the neuronal population that seems to really be expressing this channel is degenerating within the DRG. But that doesn't really tell us that CAV 3.2 is not involved in diabetic neuropathy. In fact, we think it could play a really important role. And there, I think it's important to remember that CAV 3.2, which is a T-type calcium channel and has a low threshold for activation, so it doesn't take much depolarization to cause the channel to become active. It is very sensitive to reactive oxygen and reactive nitrogen. And of course, one of the first things that happens in diabetes is that the metabolic dysfunction causes an increase in reactive oxygen in particular. And that change in the redox state within the DRG could very easily be profoundly sensitizing CAV 3.2 and leading to large amounts of calcium influx in this particular subset of A fibers. So we actually think that there's a huge opportunity here to better understand how to think about CAV 3.2 in the pathology of diabetes. And we also think that there's a good opportunity to think about CAV 3.2 inhibitors as potential neuroprotective agents early on in the disease. And we're actively working on exploring that hypothesis, working with Emmanuel Bornay's lab. So look for more work from both of our labs on that exciting idea in the future. So now what I'm going to do is to shift gears and tell you about work we've been doing on trying to better understand low back pain. And this has been led by Joe Lesniak and Khadija Mazar in my lab. Joe is a postdoc and Khadija is a PhD student. And this also is work that stems from our Precision Human Pain Network and is a great collaboration with our colleagues at University of Washington in Seattle. Christoph Hofstetter is the surgeon that we're working with, and Michele Curatolo is an anesthesiologist and pain expert who's doing the patient phenotyping in this study. So what we're doing here is we're obtaining tissues from individuals with careful phenotypic profiling and who have a history of low back pain and are having lumbar fusion surgery. And then we also recover control tissues from organ donors here at UTD. So this allows us to have control back tissue where there's no pathology in the spine, and we can visually inspect and verify that. And then we also have the tissue from individuals who've been very carefully phenotyped and have a clear history of low back pain. So we're taking two major types of tissues from this. We're taking nucleus pulposus and also annulus fibrosus. And what I'm going to do is to focus on the work that we've been doing on the annulus fibrosus, although the work on the nucleus pulposus is also really exciting. But I just want to focus on one tissue type to give you an example of how this really interesting project is progressing. So first of all, we can take the annulus fibrosus and we can do bulk RNA sequencing, and Joe has done that on a pretty large number of samples now. And again, the controls are from age-matched organ donors, and the low back pain are from the lumbar fusion surgeries. And when Joe does a comparison here of genes that are either up or down regulated in the pain samples, what we see is that there's a fairly large number of genes that are upregulated in the pain annulus fibrosis. And if there's 780 upregulated genes, how do you know which ones are potentially the most important? Well, one way that we try to deal with this issue is using this ligand receptor interactome platform that we developed many years ago. So what we do here is we intersect our human DRG expression from lumbar DRGs, which would be innervating the discs, and look at the ligands that are upregulated within the disc and pair it up with receptors within the DRG. And this helps us to focus in on some factors that we think are probably pretty important. So we see some proteases like ADAM proteases and MMP9 that are upregulated that are very important in regulating cytokine processing. And then we also see some other cytokines, FGFs, fibroblast growth factor ligands, and TGF-beta 2 that are upregulated in annulus fibrosis that have high expression level for receptors on the DRG side. So this suggests that there are almost certainly factors that are being produced by the diseased annulus fibrosis that can act directly on sensory neurons to sensitize them and maybe even activate them. So we have been doing this or exploring this by culturing annulus fibrosis as well as nucleus pulposus. But again, I'll just tell you about the annulus fibrosis data, and forgive me for misspelling fibrosis here. Joe has developed these cultures, and then what we can do is we can take media that's coming off of these cultures, so basically conditioned media, and then we can put that onto human DRG neurons and do calcium imaging and electrophysiology. So first, what happens when we do calcium imaging? So what we see with blank media is we don't really see anything. We see a nice response to high potassium. When we take conditioned media from the organ donor, healthy disc annulus fibrosis, we see a small proportion of neurons that respond to the conditioned media, but really not much happening there. On the other hand, when we do the low back pain annulus fibrosis cultures, what we see is that a fairly large proportion of neurons show a pretty robust response acutely. So that there is something in the media that can induce a calcium influx or calcium release from intracellular stores in these DRGs. So what about changing their electrophysiological status? Can it induce sensitization that would be reflected with more action potentials firing? So Mandy Schwab, who's a PhD student in Greg Dusor's lab, has been doing this work, and here again, it's very convincing. So when we look at the RIA base of these neurons, the low back pain, annulus fibrosis condition media reduces the RIA base. And also when we look at ramp current evoked action potential firing, especially in the kind of low range of the magnitude of the ramp currents, we see really big sensitization. So again, there is clearly something in the low back pain associated annulus fibrosis condition media that is sensitizing these neurons. So now one of the big things that we're trying to do is to take this condition media and use proteomic and lipidomic techniques to try to narrow in on what those factors might be. And to do that, we've developed some really exciting collaborations with people mostly in the Rejoin network, which is another HEAL initiative funded program. And Joe has been leading that work in the lab. So we'll be excited to report back on that when we have more data on trying to narrow in on what those factors are. So take home messages from the DRG side of the talk. We now have really developed unprecedented access to live human DRG here in Dallas, working with the Southwest Transplant Alliance. We've created a bunch of publicly available data sets that are available at sensoryomics.com, which I know many of you regularly use. So thank you for working with our data and for citing our papers. And also, many of these new data sets are available at spark.science, and we're going to have a huge data release this week for all the data that's going into the so-called Big DRG paper. I think we also have developed a new and very interesting view of diabetic painful neuropathy that suggests that neurodegeneration within the DRG can be a really important part of the disease. And I think that has pretty profound implications for how we might want to think about treating diabetic neuropathy. And we also have developed these kind of humanized models using tissues from surgeries and from organ donors that I'm really excited about how those can help us to better understand these very complex chronic pain disorders like low back pain, osteoarthritis, and rheumatoid arthritis. So now what I'm going to do is to transition to talking about our work on the human spinal cord. And here, I'm going to be talking about two really talented postdocs who work in my lab, Olivia Davis, who did the KCC2 work, and then Catherine Gabriel, who has led a lot of our single nucleus sequencing efforts on the human spinal cord. And this background image here is some of the really amazing xenium transcriptomic data that we have on the human DRG that Catherine and Olivia have been working on collectively. So first, I'm going to start with the KCC2 story. So KCC2 is a target that's been around in the pain field for a pretty long time. KCC2 is a potassium chloride cotransporter, and it's extremely important for GABAergic function throughout the entire central nervous system. So GABA acts on two kinds of receptors, GABA A receptors and GABA B receptors. And for GABA A receptors to function properly within the nervous system, neurons need to maintain a low level of intracellular chloride so that you'll get hyperpolarizing GABA A currents when GABA A receptors open. The major way that these neurons maintain this low level of intracellular chloride is through expression of this cotransporter, KCC2, which extrudes chloride from neurons within the CNS. Yves de Koninck's lab discovered in the early 2000s that after peripheral nerve injury, KCC2 expression is decreased, and that leads to an increase in the concentration of intracellular chloride and ultimately decreases efficacy of inhibition in the dorsal horn of the spinal cord. And essentially what that does is it opens up the pain gate and allows for more nociceptive information to flow through the spinal cord on up to the brain. So This particular finding of KCC2 downregulation is, in my view, one of the most robust and repeatedly reproduced findings in the pain literature. It's been shown across many different models. It's been shown in opioid-induced hyperalgesia. It's been shown in some inflammatory pain models. We understand a great deal about how KCC2 downregulation occurs, and we even understand that there are sexual dimorphisms in how KCC2 is regulated. But none of these studies have validated that KCC2 is decreased in the spinal cord of human beings with chronic pain disorders. So that was the goal that we wanted to accomplish in doing this study, which was published in Pain just a couple of months ago. So there were 3 groups involved in this. My group, led by Olivia, Mike Hildebrand's lab, led by Anne-Marie Dedick, and also Eve DeConnick's lab. And in Eve's lab, Samuel Ferland led the work. So what Olivia did in my lab was to take spinal cord samples that passed all of our quality control measures and use a validated KCC2 antibody and take images of these and then send them to Eve's lab to do the image analysis in a totally blinded fashion. And Eve's lab has this nice technique to look at membrane-associated KCC2. So the endpoint of these studies was looking at the amount of KCC2 in the membrane in these neurons. So I think it's pretty easy to appreciate that there's decreased KCC2 in these pain samples. We also had people who had a history of opioid use and opioid use disorder. So in our lab and the cohort we were looking at pain without opioid treatment and people who had been taking opioids and likely had opioid use disorder. And what we found is that the level of KCC2 in the membrane of neurons in the dorsal horn was pretty profoundly decreased, both in the pain samples and in the opioid samples. Most of the pain samples were neuropathic in nature, but not all of them. So I think our data supports the idea that KCC2 downregulation in the human spinal cord does not exclusively happen in neuropathic pain, but may also happen in visceral pain and musculoskeletal pain. So we also wanted to look in an independent cohort. So in Mike Hildebrand's lab, they generated a number of samples where there was no pain history, and also where there was a pain history and the individuals were taking opioids. They didn't have a no pain and opioid cohort, but again, there's a very clear decrease in KCC2 expression on the membrane of these dorsal horn neurons. So this replicated really nicely in this independent cohort in data generated in Mike's lab. So in my view, this really demonstrates that KCC2 downregulation is associated with chronic pain in humans. And I'm really excited about what is happening in this area, because there's a number of companies that have developed KCC2 positive modulators. And I'm not involved with any of those companies at all, but those companies are now taking those drugs into phase 1 clinical trials. And if those are successful, I think that we will have some phase 2 data within the coming year or so, which will be really, really exciting. And I'm very hopeful for the possibility that these KCC2 positive modulators could have really great efficacy for chronic pain in patients. So the final thing I'm going to talk about is our profiling of human dorsal horn neurons. And again, this is work that's been done by Catherine Gabriel and Olivia Davis in my lab. And one of the things that we've been really wanting to do with this project is to try to identify projection neurons in the human spinal cord. So Catherine, in collaboration with Herman Allen Poole's lab at UT Southwestern, has developed a really great technique to isolate nuclei from human spinal cord, and also to do xenium spatial transcriptomics. And we've now done this on a fairly large number of spinal cord samples where there was no history of pain, and also quite a few with a history of pain, although we've only had an opportunity so far to analyze the ones without a history of pain. So that's what I'll be telling you about today. So we see some really interesting things when we analyze this data, but I'm just going to focus on the neuronal side of things, which Olivia has been heading up. And here we have identified in the both the dorsal horn and the ventral horn what we think is a comprehensive characterization of the excitatory neurons. So these are the ones that are glutamatergic, the inhibitory neurons denoted by GABA, and their neurochemical phenotype and specific genetic markers. So that's how we have named them. So the first thing on the list here is their transmitter type. There's also a mixed type. We don't think that these are all ones that release both glutamate and GABA. We think that what they are they're transcriptomically very similar neurons, but there's a subset of them that are glutamatergic and another subset that are GABAergic, although we do see some that seem to be able to synthesize and release both glutamate and GABA. The second marker here is usually a neurotransmitter or a neuropeptide or receptor or some other marker that can be tied to the existing pain literature. And then the last one is a relatively specific gene for that particular population. And using xenium transcriptomics, we've been able to place these neurons in space. And this is just focusing on the excitatory neurons. But one thing that we were a little bit disappointed on when we saw this data is that we couldn't really pinpoint any particular population of neurons that was found both in lamina 1 and in lamina 5, which would suggest that that might be a specific population of neurons that is a projection neuron. And of course, we're interested in these projection neurons because these are the neurons that are ultimately the gateway out of the spinal cord and up to the brain, where pain is ultimately perceived. So we would really love to be able to identify these neurons to be able to mine them for potential drug targets that we could work on in the future. So to try to identify what those markers might be, we teamed up with Andy Bell from Andrew Todd's lab. Olivia did her PhD actually in Andrew Todd's lab, so she knows the group very well. And what Andy has done is to do a really nice study that was published in PNAS late last year, where they used a lineage tracing method to identify projection neurons and then classify them into subtypes using single cell sequencing. So in the mouse, we have now really nice markers for these distinct subsets of projection neurons. So the question is, can we also find these in humans? And unfortunately, when we dove into this deeper and looked to see if any of these markers were expressed by distinct subpopulations of dorsal horn neurons in humans, we really didn't find anything. So I think that the human neurons at least don't express the same specific markers that are found in projection neurons in the mouse. So how can we go about identifying these projection neurons? Well, one thing we also know about projection neurons is that they are relatively large neurons in the dorsal horn. So Olivia took on this monumental task of measuring the sizes of neurons using Noonan staining. And what she found was that about 1 to 2% of neurons in the human dorsal horn are greater than 45 microns. And we know about 1% of neurons in the dorsal horn are projection neurons. So we think that it's likely that these larger neurons are projection neurons. And in fact, when we do xenium spatial transcriptomics, we see that these larger neurons, as depicted here by these two, express some genes that we know should be in projection neurons, like the TACR1 receptor. We also see that there's some GPR83 expression in these. This is another marker of projection neurons that was identified by the Ginty lab. So when we look at these other markers across the human spinal cord, unfortunately, they're not exclusively expressed by these large diameter neurons, so we can't really use that as a marker. So Olivia had the idea that we should look in spinal cords from very young individuals, with the idea that we might be able to identify expression of these transcription factors that are expressed very early in development that ultimately specify the fate of projection neurons. So as I mentioned earlier, we have been doing recoveries from organ donors of all ages, and tragically, some very young people become organ donors. So we had some samples from individuals that were less than 1 year old at the time that they died and became an organ donor. And Olivia looked at FOX2A expression, which is the transcription factor that we know is a fate determinant transcription factor for projection neurons. And in mice, these projection neuron or these transcription factors turn off around the time of birth. However, in humans, we see that they are still on even about a year after birth. And FOX2A is very specifically expressed in very large neurons within the dorsal horn of these developing human spinal cords. So we really think that this is the clue that is going to allow us to achieve what we want to do here, because now we can identify these in these young spinal cords. Using the xenium sequencing, we can come up with combinations of markers for them, and then we can translate that into our single nucleus sequencing in the adult spinal cord, and we can build spatial transcriptomic panels that can then look at all these combinations and hopefully give us the gene expression combinations to identify all these neurons in the human spinal cord. So Olivia actually did all that work right before she left my lab to move back to Scotland, and is now analyzing all that data, and it's going to be extremely exciting to see what she finds. So the take-home messages on the spinal cord are we've now validated KCC2 as a really exceptional target both for pain and opioid use disorder in humans. With Catherine and Olivia's work, we have a really amazing new atlas of the human spinal cord that we're hoping to put out there within about the next month. And that'll include a lot of data that'll be on our sensoriomics site and on spark.science. And I feel like we're really on the doorstep of identifying these projection neurons, and that should give us a whole new way of thinking about drug development targeting the human spinal cord for pain. So with that, I'll stop, and I'm happy to take questions from everybody. But before I do that, I just want to point out the really great people that we work with here in Dallas. We have the Center for Advanced Pain Studies, which was co-founded by myself, Michael Burton, and Greg Bussor, that has now grown to 10 PIs. We are doing really great work here, and we continue to have good opportunities for students to come here. We couldn't do anything without the Southwest Transplant Alliance. And again, I want to thank the organ donors and their families, and the support from NIH, in particular for the HEAL initiative, for the Precision Pain Network, and the Rejoin Network has been absolutely phenomenal and enabled all kinds of work that I think has just been extremely interesting. And thank you for listening. And Richard, maybe you can help me to go through some questions.
Yep. Absolutely. Well, thank you, Ted, for a really great talk. And I just want to reiterate to everyone on this webinar that you can submit your questions. There's a chat box below it. It's titled questions. I also want to quickly address the question. This webinar is being recorded. Someone has asked already that, and because for people who are not able to actually join. So this video will, the link to the video will be submitted to everyone who registered to the webinar. So I'm going to start with the first questions, and harking back to the transcriptomic data that you were showing, Ted. One question that is, are you also looking at the different isoforms of, well, say, splicing variants of the genes that you are looking at?
Yeah, that's been a big effort as well. So Asta Arentrano, who is a postdoc in my lab, published a paper in Pain earlier this year using long-read sequencing, looking at splicing within the DRG. And we've really been able to expand that line of work over the last years. Over the last year, there have been quite a few advances in long-read sequencing that allow us to do much deeper sequencing on the human DRG. So we are very interested in expanding our understanding there. We do see unique splice variants. We described those in our paper for some ion channels that I think people in the pain field care a lot about, in particular acid sensing ion channels. So I think there's really exciting opportunities there for looking at variants of ion channels and other genes in human DRG.
Oh, thank you. This is a combined question too. So one of the questions is regarding the Najot model, nodules. So yeah. Do they correlate? Well, rather, shall we say, the prevalence correlate with other patient data that is available beyond what you already shown? I mean, examples would be age and drug treatment. The other one question is from same combined with this: is our specific size of the neurons that are surrounded by these nodules? It would appear that the low threshold mechanical receptors seem to be, based on your data, were the ones that were dying.
Right. So the clearest association we have is with diabetes and diabetic neuropathy, where they seem to begin early in diabetes, and then the severity and number of them becomes much greater when the individuals have both diabetes and neuropathy and painful neuropathy. We have suggestion that they might be involved in other forms of neuropathic pain, but the sample sizes are relatively small, and then we don't see a really clear pattern, for instance, with arthritis. In terms of age, like there are nodules in some DRGs where there's no disease state that we are aware of. So my suspicion is that they do increase with age, but I don't think we've looked at that quite as carefully as we probably should, and that's a good idea for us to dive into a little bit more. And then in terms of the neurons that seem to be the ones degenerating, the single cell sequencing data really suggests that it's the A fibers that are degenerating, so, and forming the nodules. So there's a population of proprioceptors that seems to be susceptible, and then there's the A LTMRs that express CAV 3.2 that I talked about. Whether or not the same population is potentially showing some degeneration with age, I don't think that we don't know that one yet. But that, again, that would be a good thing for us to look into in more detail. Ultimately, I think that this is just a matter of sample sizes, right? So we've now done about 500 organ donor tissue recoveries. And as we continue to process those, the sample sizes increase on other forms of neuropathy. It's just within the donor pool that we work with here in the Dallas area, diabetes and diabetic neuropathy are by far the most common disease that we see.
As similarly at Antibios. Thank you. So the next question is regarding, I'm going to actually add something to this too, is whether or not humanized pain models are available. So I actually would say that we actually have in vitro pain models where we test inflammatory and chemotherapy-induced neuropathy models. So I'll ask you, Ted, have you kind of developed some pain models that you want to talk about?
Yeah, we have the low back pain model that we have been working on. It requires recovering disc material from organ donors and from surgeries. So lots of logistics involved in making that happen. But I hope that the data that I've showed would convince you that that's a pretty robust model that we can use for discovery. We've done a very similar thing, working mostly with Dana Orange's lab at the Rockefeller for arthritis. So there, she's doing synovium, synoviocyte cultures, and then sending conditioned media to us, and we do it on the DRGs. Shipping the material for that has worked fantastically well, and we have some really fantastic results. So one of the things that we're trying to do now is to work with leading experts in areas of pain like arthritis, where we don't really have the expertise. And this has been one of the amazing things about these HEAL initiative programs that have been put together. So, the way that we've kind of integrated our lab with the folks in Rejoin, mostly Dana's lab and Anne-Marie Malfay, and also Martin Lotz at Scripps, has just been amazing and has led to all kinds of really great new projects that I think are going to tell us a lot about molecular mechanisms of pain in these very, very prevalent conditions.
Thank you. So the next question is about proteomic approaches and whether or not you have considered applying actually a multiplex approach that's combined with patch clamp.
So we haven't done it with patch clamp. What we have been doing is laser capture microdissection proteomics. So Stephanie and Ali Berry have been leading that. So Ali, so this has been a collaboration with Manuela Schmidt's lab in Austria. We had a paper published in Pain on bulk proteomics on human DRG that Ali led, and then we've been doing this laser capture microdissection work that Stephanie and Ali have been working on together that has really worked fantastically well. And then we also developed a new collaboration with Cherry Ho at Johns Hopkins, looking at a cancer pain population, human cancer pain population, where she's doing autopsies, and she's a neuropathologist and doing some really neat stuff related to laser capture microdissection proteomics. So that I think all of that is really coming along amazingly well now. And I know some of the other labs in the precision group are also doing proteomic work, like Rob Giro's lab and Juliet Marighi. So the proteomic technologies have been a little bit behind the sequencing area, but I think now we're getting to the point where we can reliably measure ion channels and G protein coupled receptors using the newer mass spec machines and these more advanced tissue isolation or protein isolation protocols. So it's getting to be pretty exciting times for proteomics.
Thank you. So what's known about the role of microglia in terms of KCC2? And are any of the ongoing clinical trials to modulate microglia activity? Is that a means to be able to affect KCC2 for pain?
Yeah, sure. So I guess the story on that from the preclinical literature is pretty clear. There's this idea of, and a lot of data behind it, of BDNF release from microglia acting on TrkB on neurons that express KCC2 and causing downregulation, although that seems to be something that happens more in male spinal cord than in the female spinal cord, at least in mice. In humans, we're just starting to look at microglia and astrocyte activation, and I think that's something that Catherine is analyzing right now in her work on single-nucleus and spatial transcriptomics on these neuropathic pain spinal cords. So we need a little bit more time to get all that data analyzed, because it really is a ton of work to put it all together, but I'm pretty excited to see what we're going to find as that moves ahead.
Thank you. So here's a question about NAB 1.7. I think, and in the spinal cord. So I know if you've reported this. So the question is, regarding the dorsal horn labeling for NAV 1.7, and it's just basically asking if it's expressed postsynaptically, and have you seen, what have you seen in terms of the spinal cord neurons?
So we see in every transcriptomic method that we have used, very broad expression of NAV 1.7 mRNA in spinal cord neurons, in both the dorsal horn and the ventral horn. The protein labeling is harder to decipher. So first of all, the protein labeling of the incoming afferents into the spinal cord is intense. So making out, what's happening potentially postsynaptically is challenging. Having said that, Stephanie has done a lot of work to try to understand that, and I think that the conclusion that we've reached is that where we do feel confident that we have seen NAV 1.7 in dorsal horn neurons, at least, is in the axon initial segment of a population of neurons in the dorsal horn, and also in the axons that are crossing the midline. So, we presume that those are projection neurons that are crossing the midline and then, are going to form the beginning of the spinal thalamic tract. So the staining along the axons of those neurons does not look anything like what the staining looks like on afferents. The density of 1.7 in those neurons is not even close to the same, but 1.7 could be playing an important role in the axon initial segment in a subset of neurons, because we do see staining there. So I think it's still a mystery what the function is exactly of spinal cord NAV 1.7, but from our perspective, in terms of mRNA expression, it's just as clear as can be that the many neurons within the spinal cord express NAV 1.7 mRNA.
Thank you. The next questions comes, first, a great talk on terms of, as well as looking forward to your big DRG paper, but the question is about chemotherapy induced peripheral neuropathy, whether or not you have been recovering from organ donors with CIPN. And the second part to this, I don't think this is with regard to the CIPN, but nonetheless, if you've seen anything, it's a question about expanding on the sexual dimorphism that you've been that you referred to. I think with regard to spinal cord.
Yeah. So on the CIPN question, that's a rule out for organ donation. So no, we have not. Now, we have this wonderful collaboration with Pat Doherty at MD Anderson for these thoracic vertebrectomy DRGs, and many of those donors or many of those patients do likely have chemotherapy neuropathy. It's just that they're extremely complex, and it's very hard to know what is the source of their pain, because they also have profound disc degeneration and nerve compression. So I think that there we would like to understand that better, but it's a challenge to figure out how exactly to get samples that would help with that. But as you already mentioned, Richard, you can do in vitro models, and I know that your in vitro model shows pretty profound changes in the DRG neurons with exposure to these chemotherapeutic agents. So I think that there's other ways to potentially address it. In terms of the sex differences in the spinal cord, we do see sex differences. That's again something that Catherine is working a lot on, and the differences aren't really in the neuronal populations or in the gene expression within the neuronal populations. It's in the astrocytes and the microglia. So there are particular subsets of astrocytes that are overrepresented in males or females, and vice versa, and the same for microglia. And they seem to have very distinct functions. So I think that when Catherine is done with all of that work and we're ready to publish our human spinal cord paper, people will be excited about those sex differences. And then, obviously we have to try to understand what they actually mean.
Thank you. So the next question is regarding satellite glial cells and the nodules. Have you been looking into that?
Yeah, so if you look at the Nature Communications paper that Stephanie published earlier this year, we did spatial sequencing on these samples, and we got a lot of data in there about the cells that are within the nodules, and we deconvoluted it, found different types, so what are likely different subtypes of satellite glial cells. And then following up on that, now we have a lot of single nucleus sequencing from these, and we can do a better job of deconvoluting it based on that. So I think that there are some distinct phenotypes of the satellite glial cells that form the Nejet nodules, and we're trying to understand now what those satellite glia release that might be causing the incredible sprouting that we see happening within the DRG.
Thank you. So I'm going to note, but there are a lot of questions still coming in, and I know you have some. I mean, you've been very gracious in going over the time, but you do have a hard stop at this point. So I'm going to try to focus on questions I haven't asked yet. First of all, can you comment on the differences? Well, I mean, the difference in using the iPS-derived sensory neurons to model neuropathic pain, in comparison to, say, primary sensory neurons, which we both work on.
Yeah, sure. I mean, I think the iPSC neurons have a ton of potential. There are some differences. I think that understanding those differences will improve the protocols for the iPSC neurons. We've been kind of actively working on that with anatomic. I know there's other cells that are available, and I'm sure other groups are working on it in different ways. So yeah, my view is that this is a technology that you can easily understand the pluses and minuses, because that there's a lot of high throughput data, sequencing data, and electrophysiology data that's available to allow you to evaluate, if it's appropriate for certain targets and underlying mechanisms that you might be interested in. And then also, we are getting a better understanding of why the differences exist and how we can try to make the iPSCs better. And I think that eventually we're going to get there, and I don't think it's going to take all that long. So, that obviously would be a real breakthrough. There's also this cell line that Derek Molliver's lab recently developed that looks pretty exciting to me. So there's, I think there's a lot of really neat work that's happening on trying to develop other ways to kind of humanize or have humanized models for the peripheral nervous system that can be used for mechanism and understanding and drug development.
Thank you. The next question is, well, could you comment on the relationship, what you think the relationship is between the transcriptomic signals that you're picking up and proteome or protein expression?
Yeah, so one of the things I've been interested in my entire career is translation regulation and how it contributes to neuronal excitability. There are disconnects, so I don't think you should look at transcriptomic data and say, this gene is very highly expressed, therefore it's going to have a ton of protein. But there are also major gaps in the kind of like visibility of certain types of proteins with mass spec applications. So I don't think we're quite there yet to be able to, put a mathematical model to this. But being able to do so in the future would be a big breakthrough. So I think there probably will be genes where there'll be a very direct correlation between the RNA amount and the protein amount, and there'll be other genes where there'll be a massive disconnect. And it'll be important to understand what those mechanisms are. And what I would guess, based on what we know about how translation regulation contributes to nociceptor excitability, is we'll see that many of those genes that do have kind of an anti-correlation will be important for sensitization, because what happens probably is that those genes get turned on with pre-existing pools of RNA and are translated, and that they rapidly lead to changes in excitability.
Okay, thank you. So the next question is regarding the T-type data that you presented and the decrease that you see in diabetic neuropathy. So is this a decrease seen throughout the disease, or is something that progressively develops as the disease progresses?
We don't really know that because it's hard to really have the samples that you have like the precise timing on them, right? So our hypothesis is that these neurons are more susceptible based on the single nuke sequencing, and that it happens early in disease. I think late in disease, there's very good evidence from what we've been doing that the channel is mostly lost. So I definitely wouldn't take the approach that already failed in the clinic, of trying to inhibit T-type calcium channels for diabetic neuropathy late in disease. But again, I think there's a lot of opportunity to go after that mechanism at the early stages of disease. If T-type inhibitors are safe, that's a key issue, obviously.
Thank you. Given the time, I'm going to ask 3 more questions, even though there's more.
Okay.
But hopefully...
I could probably go till about 25 after the hour, and then I really need to go.
Okay. All right. That's good to know. Thank you. So the question is, have you been, using the conditioned media from the annulus fibrosis? So have you looked at some other conditioned media from other patients and been, collaborating or doing it yourself on stimulating on the DRG neurons?
Yeah, we've been able to do it from other types of samples from the low back, or other types of cultured cells from the low back samples, and then we've been doing this collaborative work mostly with Dana Orange's lab, looking at synovium, cultured synovial samples from rheumatoid arthritis and osteoarthritis, with very exciting results across the board. So I think that this is a really great area to go for mechanistic understanding and eventually for drug testing.
Thank you. So here's a question again about nodules, as whether or not you see differences based on the level of the DRG, so lumbar versus cervical and thoracic.
Yeah, that is something that we're trying to look at in more detail, because one would imagine that at least earlier on in the disease that it would be almost exclusively happening at the lumbar levels, because when the neuropathy is developing, it's very clearly link dependent. So that is something that Stephanie and I have been working on. Don't have a lot of data on that yet. We have some evidence that we published in the Nature Communications paper that at samples that are coming late from disease, we see it in thoracic DRGs, but the neuropathy does spread to other levels as the neuropathy gets more severe, and these cases were probably pretty severe.
Right. Thank you. So there's a question about the process of procurement with organ donors. So some of these people prior to passing away, they of course have been in treatment. They've certainly been on potentially on pain medications such as fentanyl. So have you had any? Do you have any data, or what do you think might be happening in terms of that impact on the DRG, specifically on the DRG and the spinal cord?
That is true. Not true in every case, but true in many cases. We have, for instance, data with individuals that had a long history of opioid use versus controls, and we see profound differences in KCC2 expression, for instance. I do think that if you're going to do this kind of work, like you have to become comfortable with the messiness of human disease, number one, and also, the things that you just have to accept are going to happen during the process of recovering the tissues. So you know, the organ donors are going to be on life support. There's nothing we can do about that. The surgical samples we get give us a good control, but you know, we don't ever get lumbar DRGs from surgery, because there's no surgery where people are removing lumbar DRGs. There are thoracic DRGs and cervical DRGs. So one of the things that we have done is to compare the thoracic DRGs from the thoracic vertebrectomy surgeries to thoracic DRGs from organ donors, and we don't really see any differences in the transcriptomic profile of the cells between the organ donors and the surgically resected DRGs. Now we do see some differences that we think are related to neuropathy and nerve compression, but you know, whether the organ donation process changes the transcriptome of the DRGs, we don't have any evidence that that is the case.
Yeah, I would agree, and of course, you know, that's also our experience. But it's also true that you can't control for everything. So the fact that this is human data, I think, trumps everything.
Yeah, and you know, the other thing that I at least have learned in doing all this is, which of course maybe we should all know. I mean, people having a single pain condition, once they develop pain, is kind of rare. We see routinely people have knee arthritis and low back pain, which came first. Probably we won't have in the medical history, but we will have a fair amount of detail about both of them. We see people who have neuropathic pain and migraine. We see people who have visceral pain and arthritis. Yeah. I mean,
yes.
Human disease is very complex, and if we're not going to make an effort eventually to understand the complexities of human disease, then what are we doing?
Right. We are getting close to the 25 after the hour, so I'm going to ask this one last question because it could take some time. Answer about what's the role of non-neuronal cells in neuropathic pain?
Oh, well, I mean, they definitely have a role. You know, we see the satellite glial cells, for instance, associated with the Nejet nodules. We've seen fibroblast changes within the DRG. We see immune cell changes within the DRG. We've been doing this work with Peter Grace on B cells and the DRG. One thing that I'll just kind of boil it down to kind of one idea. So what I expected when we got into this is that I thought that the DRG neurons would just change their transcriptomic profiles like crazy. And, you know, it's all about the neurons, but, you know, I've always been a neurocentric person, and that's just not what we see. We do see some changes within the neurons, but, you know, it's not, they're not huge. They might be really, I mean, I'm sure that they're important, but the things that we see that really seem to be changing are the other cell types. So I think it's important to remember that these neurons at their basal state express receptors for like anything that can be released by these other cell types, really. And those things coming from those other cell types are almost certainly, you know, creating ongoing drivers of changes in excitability within the neurons. And some of it is probably driven by things that we can't possibly see when we're just looking at the DRG, but I'm more and more convinced that there is pathology within the DRG that's associated with these diseases that gives us good opportunities for intervention.
Thank you, Ted, very much. And, you know, I apologize to anyone who I've not asked the question, but we appreciate that the interest, the fact that you stayed longer, both to the people in the audience as well to you, Ted. And I guess it certainly speaks to the interest and the advances that you and your collaborators have been making in this field. So again, I want to thank you for your.
Thanks, everybody.
Yes, thank you to everybody, and you know, please join us next time we have a pain webinar. And thank you.